can turn off one mic and use the other still working. Great. So now it is my great pleasure to introduce our next speaker, who is an exceptional rising star, Dr. Leonardo Tozzi. Dr. Tozzi is an academic staff scientist and researcher in the Stanford Center for Precision Mental Health and Wellness. He received his PhD from Pisa University in 2013 and his MD from Pisa and his PhD from Trinity College London, Dublin in 2018. He joined us at Stanford in 2018, and since then he's done some extraordinary work. So he joined us to lead a really important study that was the first time we used human connectome imaging in depression, and he will include some of those data in his presentation. He's also led as Associate Director the Computational Neuroscience and Neuroimaging group in our center, which serves to integrate data across our projects, and I'll talk more about that in a later session today. So Dr. Tozzi aims to develop quantitative biomarkers for psychiatry that are reliable, interpretable, and useful for treatment selection. So you'll hear more about this today and how he's integrated a very large set of data to generate some very novel outcomes for depression. So he will present on personalized brain circuit scores, linking biotypes, symptoms, behavior, and treatment outcomes. Join me in welcoming Dr. Tozzi.
Thank you very much for the very kind introduction and for having me here today. It's really a great pleasure and an honor to be talking at this fantastic symposium. So today I'm going to present the results of a study we conducted recently on a large cohort of participants with depression and associated disorders. And the important thing of the study is in this large cohort, we used what we call personalized brain circuit scores, which means measures of brain circuit function, to group these participants into separate subtypes based on their brain biology. And I'm going to show today that these subtypes then that we call biotypes differ in terms of their symptoms, in terms of their performance on computerized behavioral tests, and also in terms of their treatment outcomes across multiple treatments. I don't have any conflicts of interest. So, you know, we've heard this many times, even today. One of the challenges we face today in psychiatry is that we are running most of our studies or making our clinical decisions based on symptom-based categories, right? For example, major depressive disorder. And these are notoriously heterogeneous. And the problem is that by doing so, we risk conflating people who maybe are actually different into one big bin and put them all together in the same bucket. So precision psychiatry and in general, many research initiatives have tried to tackle this problem. And the idea here is to use sources of information that are not symptoms and that can come from many different domains. For example, you know, you have some on the slide: genetics, brain activity, physiology, inflammation, behavioral biomarkers, and also backgrounds, life experiences, and so on. And the idea is that by using these other sources of information, we can use data-driven techniques to reach a more homogeneous grouping of these participants. And the idea is that when we get to this more homogeneous grouping, then we can make more informed and better treatment decisions. So in the ideal scenario, some groups will respond better to a treatment compared to others, and so on and so forth. So what I'm going to focus on today is this type of framework, but specifically focused on measures of brain function or brain activity. So, and this work really builds on previous work on this type, on using brain function to define subtypes of mood disorders, right? That have been called biotypes in the literature. And this is the term I will use here. So what do we want our ideal biotype to be like? So first of all, we should be able to have a personalized assignment to a biotype, right? So if a patient walks in, we should be able to allocate that one person to a biotype or another one. Second, a desirable property would be that we would be able to also express how dysfunctional the brain activity of a patient is compared to some sort of norm. Like if a brain circuit is altered, how altered is it? Is it a lot? Is it a little bit or not at all? A third desirable property is that we want our biomarkers to be interpretable. And by interpretable, I mean to be tied to some neuroscientific interpretation of what we know of how the brain works. So something we can, also interpret mechanistically or use to develop new hypotheses or test new treatment and new target and use potentially as target for treatment in the future. Then ideally, we want our biotypes not only to differ in terms of brain, but also to differ on other units of analysis. These could be symptoms, but also, for example, computerized tests of behavior. And finally, we want our biotypes to be useful for treatment selection. So, for example, one biotype should respond better to a treatment compared to another one. So this was the goal of our study, and the biotypes I'm going to present to you today satisfy these criteria. So in the current study, we leveraged a large data set coming from the pooling of many studies that have been conducted in our center. It's composed by 801 untreated participants with this depression, but also other related disorders, and I'll show you the breakdown in a minute. Then we deployed a circuit quantification pipeline that we developed in the center that is able to provide, interpretable and normed and personalized measures of brain circuit dysfunction for each of our participants. Then we applied data-driven clustering algorithms to identify what we call these 6 biotypes, which I'm going to show you today. And these are groups of participants, clinical participants, grouped based on their brain biology. And finally, to validate these biotypes, we proceeded to check whether they differed on information collected from other sources. That means self-reports, questionnaires, but also computerized tests of behavior, and also if these biotypes differed in terms of their clinical response to both pharmacological treatment and behavioral therapy. So I mentioned that this data comes from the pooling of several research studies that have been conducted in our center for the past years. So you can see here the 4 main studies that comprise this cohort. So the top 2 studies, the Human Connectome Project for Disordered Emotional States and the so-called RAD project, are cross-sectional studies that include a large number of participants with depression and also anxiety disorders that have been scanned here at Stanford. And the bottom 2 studies are clinical trials. So on the left, you see the iSpot-D trial, which involved administering to treatment-naive patients escitalopram, sertraline, or venlafaxine, and then seeing how they would do after the treatment. And then we also have the ENGAGE study, which was a study evaluating a combined behavioral intervention for treating depression and obesity. So here we have large cross-sectional data sets, but also the possibility to study how the biotypes we identify respond to both pharmacological and behavioral treatment in time. Regarding the diagnosis of this cohort, you can see here as pie charts the disorders. Not all participants met the meaning criteria for DSM, traditional disorders. You can see the most present disorder in this cohort was major depression, followed by several comorbid anxiety disorders, and also a small number of participants also suffered from OCD and PTSD. And then this is what I was saying before. So 237 participants from the large data set also went on to take part in one of these 2 clinical trials. So we were able to have access to data of around 50 people for each possible treatment arm. So as you can see, there were 3 pharmacological arms, escitalopram, sertraline, and venlafaxine, very common first-line antidepressants. Then 1 behavioral problem-solving treatment arm of a treatment called iCare, and then 1 usual care arm. So I want to talk a little bit more in detail about our brain circuit measures extraction pipeline, both because neuroimaging is my passion, but also because I think it's important to dive a little bit more in detail in this aspect of the work. So To make our findings more interpretable and to obtain a number of biomarkers that was manageable for then our data-driven clustering procedures, we decided to focus on the function exclusively of 7 brain circuits that have been widely reported to be affected in depression, mood disorders, and also associated disorders. These are the default mode circuit, the salience circuit, the attention circuit, the circuit that underlies the process of sadness, so a negative affect circuit. Another circuit that underlies the affective processing of threat, so another form of negative emotion. The positive affect circuit or reward circuit, which underlies the processing of positively affective stimuli. And finally, a cognitive control circuit, which has also been shown to be disrupted in depression and associated disorders. So for each of these circuits, we use a specific task or type of stimulus in the scanner to probe their function. So as you can see here, for the first 3 circuits, we probe their function in the resting state or intrinsic state, where we just collect brain activity in the absence of any stimulus. But then for the other circuits, we leverage some tailored tasks that are designed to probe the activity of that specific circuit. So for example, for the negative SAD circuit, we have a task that involves the presentation of sad faces. For the threat one, we have a task that involves the presentation of threatening faces, both consciously and unconsciously, because we believe there might be different ways in which a circuit is activated in these 2 conditions. For the positive affect circuit, we use happy faces as a socially rewarding stimulus. And finally, for the cognitive circuit, we use a go, no-go task where people have to inhibit their pressing to a no-go stimulus. We focus specifically on a very targeted area that we identified to be involved in these processes by the analysis of large meta-analytic results. So these are regions that are both known to be involved in the processes I'm talking about above, but also to be disrupted in depression and anxiety. So it's kind of an and of these 2 criteria. And this allows us to keep the features we extract for our imaging data to a minimum, so that then when we feed them into our data-driven techniques, we don't lose control of our feature space. And for these measures, we compute, from these regions, we compute measures of both activation and connectivity. And by that, I mean in the resting state, as you have seen in previous talks as well, we compute functional connectivity or the synchronization of fluctuations of the bold signal across pairs of regions. Whereas in tasks, we compute both activation, so response to the stimulus of the region, and also functional connectivity in the task, meaning how does the functional connectivity change in response to a stimulus between pairs of regions. And these are summarized here, in a cartoon representation. And you can see that by doing so, we can identify a smaller set of features, which then provide the input to then our successive step of analysis. So for example, for the default mode circuit, which is the one I'm showing now, we can calculate, we select 4 regions and we can calculate the functional connectivity between each pair of regions, right? So we focus on some specific connections that we've shown are specifically impaired in depression and anxiety. And we use each one as a feature for our successive then data-driven clustering process. So by doing so for each of these circuits, we can extract a total of 41 brain circuit scores per participant. And we call these regional brain circuit scores. So there's one more step that's missing here is how do we know if one brain circuit score is elevated or within the norm for a certain type of feature? So when is amygdala activation in response to negative faces too high, right? So we leveraged a data set of healthy controls, which we used as a norm to define how much the single circuit score for each participant deviates from the norm in terms of standard deviations from the normative values of healthy people. So for example, here, you have the example of 2 clinical participants, right? On the y-axis, you have the dysfunction of the individual circuit score relative to a healthy norm, where 0 corresponds to the mean of the healthy controls, and 1 corresponds to 1 standard deviation above or below the healthy controls. And you can see that we can express each person's brain circuit scores as the deviation from the healthy norm. So for example, in this case, the red person here has a particularly high brain circuit score in the attention circuit, shown in yellow, as you can see in that massive peak, whereas the blue person in that score, in that circuit is near 0, so has no dysfunction, and so on and so forth. So we can calculate personalized measures of brain circuit dysfunction relative to a healthy norm for each of our participants. And then we used this as input in a hierarchical clustering procedure where we grouped together people who showed a similar pattern of circuit dysfunction. So I'm going to gloss over a little bit, for the sake of time, over our clustering procedure, but I'm very happy to answer, technical questions in the Q&A about how exactly we perform the clustering and what algorithms we use. But, now I will present the biotypes we identified after the clustering procedure. So the way I'm going to do this is I'm going to show as a color code, 1 color per each of the circuits we have identified at the bottom. And I'm going to show in color the circuits for which the biotype on average had a dysfunction that was higher than 0.5 standard deviations from the norm. And we use this criterion to kind of find the most salient disruptions of each biotype. So for example, here, this first biotype was characterized by elevated brain circuit scores in the default, attention, and salient circuits. And you can see that highlighted in the 3 colored bars. We decided to adopt a naming convention for these biotypes by using as a capital letter the circuit that was dysfunctional, and then as a small letter whether it was connectivity or activation that was dysfunctional, and then as a plus or minus sign the direction. So for example, this circuit is characterized by hyperconnectivity of the default, attention, and salient circuits. And we also use a cartoon representation like this, where the spheres that are large spheres signal hyperactivity or hyperconnectivity, and thicker lines also signal hyperconnectivity. So this was the first biotype we identified, characterized by hyperconnectivity in all the resting state networks. The second biotype we identified was characterized by markedly lower connectivity in the attention network. Then we identified a third biotype that was characterized by hyper-response and hyperactivation to affective faces, both threatening and both positive. Then we identified another biotype that was characterized by markedly higher activation in response to our cognitive task. One that was characterized by lower activation in response to threatening face, lower connectivity, sorry, in response of threatening faces, and also lower activation in response to our cognitive task. And finally, one biotype which really didn't show much deviation from the norm across any of our brain circuit measures. And we decided to use the letter X to express the fact that it wasn't neither higher nor lower, so neither plus nor minus here in our nomenclature. So we went through several quality checks to make sure that these biotypes were robust. Again, I can talk more in detail in the Q&A here. I'm just showing one. We repeated our clustering procedure in 2 random splits of the data, and then we observed that we see very, very similar, if not identical in most cases, profiles of circuit dysfunction across clusters. So that reassured us. But we also adopted many other quality checks for that, so I'm happy to talk about that later. So now moving on to the clinical validation. So we proceeded then to study whether each cluster or each biotype was different from other biotypes. So to the participants belonging to other biotypes in terms of their symptoms, in terms of their cognitive performance in behavioral computerized tests, and also treatment response, right? And here I'm going to show these results as a very high overview as radar plots, and I'm going to use the symbols that you see at the bottom where you will see as a red circle a biotype that had comparatively higher symptom severity or treatment response compared to participants in other biotypes. Or, and then I'm going to use these other smaller symbols just to signal some of the quality checks we did to make sure that our results were reproducible. So we replicated some results in a split half replication, and we also showed that some of our results even replicated when leaving one site out entirely from the analysis and then finding the results both in the held out site on its own and then in the bigger data set. And this is signaled by a small triangle there. So moving to the first biotype, the one that I said was characterized by hyperconnectivity in resting state networks, we didn't find that it was different from the other ones in terms of symptom severity. We did find that it had a significantly higher behavioral dysfunction in tests that involved explicit emotion recognition, but also executive function and attention. And finally, we also observed that this biotype responded particularly well compared to other biotypes to the behavioral treatment. Moving on to the second biotype, interestingly, the biotype that was characterized by lowered functional connectivity in the attention circuit also was the one that performed worse in attention. So I thought it was interesting. It didn't have many other differences compared to other biotypes. Moving on, the biotype is characterized by hyperactivation, both in response to sad faces and to positive faces, was also the one characterized by the highest anhedonia and ruminative brooding compared to the other biotypes in terms of self-reported symptoms. Then interestingly, this biotype that was characterized by higher activation during a cognitive task involving cognitive inhibition was characterized not only by deficits in cognition, especially in cognitive control, which makes sense because that's exactly what the task was probing, but also was characterized by many differences in terms of symptoms, including negative bias, threat dysregulation, anhedonia, and also anxiety. And also this biotype was the one that responded best to venlafaxine compared to the other biotypes. Moving on to the last two biotypes, the biotype that was characterized by lower connectivity lower activation in the cognitive control circuit and lower connectivity in response to threatening faces was the smallest by far. So it's not surprising here that we didn't find many really clinical results that make it stand apart from the other biotypes. And finally, the biotype that was characterized by relatively comparable brain function to the healthy controls was also not characterized by many differences in terms of symptoms and treatment response, but also was characterized by higher reactivity to implicit threat on computerized tests. This biotype was also relatively small. And when you see, by the way, that in the treatment radar, there are no lines, it's because we simply didn't have enough sample size to test whether this biotype did better on treatment compared to others. And the reason for this is that sometimes, because not all of our participants went on to the treatment arms, some biotypes, unfortunately, were not well represented in the treatment arms. So that's something that in the future we really would like to explore. So then we asked, okay, do these biotypes map to traditional DSM diagnosis, right? Is there some kind of overlap there or not? Well, it turns out not so much, but maybe, there is to some extent for major depressive disorder. So there was a significant overrepresentation of major depressive disorder patients in some of the biotypes compared to others, in particular, the three biotypes I'm showing on the figure. So the one characterized by hyperconnectivity in resting state networks, the one characterized by hypoconnectivity in the attention network, and the one characterized by hyperreactivity in response to emotional phases. Those contained significantly more patients diagnosed with MDD compared to the other biotypes, which proportionally contained less. Regarding the other comorbid diagnosis, however, we didn't see any significant difference. So to conclude, I've shown the results of a study where we took a large cohort assembled across large studies, both treatment trials and cross-sectional trials, to make up 801 untreated participants with depression and related disorders. I've shown how we used a pipeline to derive personalized and normed brain circuit scores from these data. And I've shown that we can use data-driven techniques to generate and to identify 6 biotypes from this data. And then I've shown also that these biotypes are not only different in terms of imaging, but they also are different in terms of symptoms, performance on cognitive tests, and the response to pharmacological and behavioral treatment. So in the future, what we would love to do, of course, is to study, and to use this information, to use this pipeline to assign people prospectively to a treatment. For example, we've seen how interesting the results were for the biotype characterized by hyperactivation in the cognitive control circuit. And one of the nodes of that is the DLPFC, which has been talked about regarding TMS and so on. We also are at the moment conducting studies in the center that target specifically some of these circuits. And we really would like to see how these biotypes perform longitudinally when we use a treatment that is supposed to target the circuit that is dysfunctional in that biotype. So with that, I really want to thank my colleagues and all the staff at the Center for Precision Mental Health and Wellness. And thank you very much all for your attention. And I'm open to any questions. Thank you.
Thanks for a wonderful presentation. Maybe we'll take 1 or 2 brief questions, then we're going to have an impromptu short break for everyone to stretch their legs and then the discussion session. I want to welcome some of our supporters of our research who make it possible, who've joined us for this afternoon session as well. So, questions?
I think there's one there. The microphone is coming.
Yes, thank you for the great talk.
Thank you.
So for assigning individuals to these biotypes, you require resting state and task-based fMRIs. Can you imagine using some other type of imaging, which is more inexpensive, that allows you to assign individuals to a biotype?
You mean other types beyond resting state and task?
Yes.
Oh, yeah. So that's a great question. So here we used, as you said, tasks and also, resting state data. But also one thing we did, I don't know about how this would change if we had more modalities like structural, diffusion, you name it. For sure, this is doable, and possibly we would identify new biotypes. But of course, then the problem becomes also, the more features you add, the more messy it can potentially get. One thing we did look at, though, is how does the biotyping change if, for example, we take the tasks away? Because that's usually, what people ask us often is like, do we need the tasks, right? Or is the resting state sufficient? So there's interesting things that happen if you take the tasks away. One of the things we see is that you still do get some biotypes that differ on, say, symptoms and even treatment response. The thing that happens is that these biotypes don't start not passing some of the quality checks that we implemented in terms of, for example, beating the clustering you would normally expect from random data, for example. So we think, like, in a way, the features we have are important, at least in defining some biotypes, because as you can see, like some of the biotypes that are differentiated in terms of task responses also have some treatment outcomes that are particular to them. So I would expect if you add, then it's probably good and you'll get more granularity, but there's a point where if you take too much away, then your clustering starts getting worse, basically. I don't know if that answers your question.
No, definitely. That was also, my question was more on the side of, can you take, say, the task based away? And you answered that already. So
I expected
you can,
that
but then the quality checks
Thank you.
Partially. Thank you.
Thank you. Yes.
Go ahead.
Hi, thank you for the wonderful talk. I have a quick question about your clustering technique, because there's so many common
Yes.
clustering algorithms, and I'm a bit curious what you
Yes.
arrived at and
Yes, that's
what performed well.
Yeah, that's a great question. So in the end, what performed really well for us was a hierarchical clustering. But the thing that was most surprising, perhaps, was the choice of distance metric. That was really what made it for us. So as you know, most of the time by default, one would go with something like a Euclidean distance or something like that. But for us, I found that we were inspired by a previous paper that applied a similar approach, but to different data, and we ended up using. Let me see. We ended up using the correlation as a distance metric between the profiles of this function, and let me show you what I mean by that. So here. Sorry, now I have to. Oh yeah, here. So as you can see here, the two like mock patients here, they have a profile of circuit dysfunction, right? And you can calculate the correlation between these two profiles of circuit dysfunction, and that can give you a distance between individuals. And the best success, like actually this was the breakthrough actually to make the clustering work in this case, was to use this as the distance. That's when we started seeing a structure appear. Because when we were using something like the Euclidean distance or other kind of more linear distances, we weren't really seeing that much structure. So I'm happy to talk more in detail about this and maybe tell you all the checks we did to let's say, quantitatively demonstrate that our clusters are quote, unquote, real, right? There's tons of indices and things like that, but the breakthrough to passing all those checks was when we landed on this type of distance that took into account the entire profile of this function and not just the individual deviances in each circuit. Yeah.
Thank you.
You're welcome. I think, oh, sorry, we have time. Or,
Do we have enough?
I saw another hand there, but I don't know.
Maybe we
not.
save it for the discussion. I can see there'll be a great discussion.
Thank you.
Thank you